Millions of animals are euthanized for research and drug development every year. Gelomics is eliminating the need for animal testing in pharmaceutical drug development with their 3D tissue culture solution. So Christoph, can you share how this technology works and compares to current processes?
Preclinical drug development still heavily relies on animal and cell experiments. In 2021 alone, over 200 million animals were euthanized and used for research purposes. Yet, over 90% of drug candidates that successfully pass through many years of studies in both cells and animals will fail when tested in human clinical trials. This is of course a massive problem for the pharmaceutical industry, as the failure of one single drug candidate can exceed $1.3 billion in costs. We combine cutting-edge laboratory equipment with specialized materials that are designed to interact with human cells on a microscopic level, and lastly with AI machine learning algorithms to grow human living tissues in a petri dish. And this technology allows us to generate small little beating hearts, microscopic little livers, or even patient-derived tumors that resemble the disease that we see in the patient as well. So by using these simple models, pharmaceutical companies can test their drugs much more effectively. They save a lot of cost, they save a lot of money, of course, and they reduce the reliance on animal testing, all by creating more predictive data sets that allow you to progress your drug development quicker and safer than ever before.
So the living tissue that you are creating acts, feels, behaves, and looks like human tissue.
That's exactly right. So by combining all of these materials and approaches, we for the first time are able to generate lifelike little tissues at scale and at the quality and reproducibility that the pharmaceutical industry really needs.
So how would this solution fit into existing processes?
So currently, in the early preclinical drug development pipeline, drug candidates are extensively tested using cell-based models. In these cell-based models, cells are typically taken out of a human tissue and placed on a stiff two-dimensional plastic surface, a petri dish. This, of course, is for the cells a very artificial environment, and they will start behaving up naturally. And of course, the data you generate using such systems rarely predicts how human biology really works. Our system, on the other hand, replaces the cell-based assays with tissue-based assays. Tissues are more complex, and the drug responses are also more predictive and more similar to what you would observe inside of the human body.
You mentioned artificial intelligence. How are you using AI in your product offering?
So AI is absolutely core to what we do as well. And that's because tissues are much more complex than just cells. There's more factors that actually influence the growth of tissues, their drug responses, the analytics of all the complex data sets. Previously, there was a trial and error approach. There's many factors that influence the growth of these tissues and their development. You had to try many different factors. It would take years, cost tens of thousands, maybe hundreds or even millions before you would get to a meaningful result. By using machine learning models that are trained on experimental data sets, we can really fast-track that entire process and simplify it, streamline it, entirely integrate it from the very early stages of drug development to later stages as well.
How does your solution compare to other products on the market?
Currently, market-leading products are actually grown in mice. They're generated by injecting tumor cells into a mouse. The mouse then grows a tumor, but of course this is not only ethically very challenging because it actually contributes to the use of animals in research rather than reducing it, but it also leads to huge problems with reproducibility, scalability, and also the ease of use of these materials. So what we are trying to do is to overcome these challenges and produce a product that is highly reliable, reproducible every single time.
How do you stay competitive given how rapidly changing the healthcare space and industry is? And how do you protect your intellectual property?
Staying competitive is, of course, a very challenging topic. And our approach to this is that we continue investing into research and development. Even with a product on market, we continuously improve our services, our product offering to really address new challenges that might arise in both drug development and also in medicine, and also stay ahead of the competition.
So where to from here? What's next for Gelomics, and what is your scaling plans?
So we have really worked on standardizing our manufacturing processes and also getting those ISO certified. So very close to having a quality management system in place that is recognized internationally as the leading standard. And of course, with that, we are looking forward to scale our company even more, penetrate into new markets, both from an application perspective, but also from a local geographic perspective. So for us, the next steps is really to make it big in the US and also in Europe, and partner with more pharmaceutical companies and bring our solution into their day-to-day workflows.